system. Okay, so now you have to go over autonomic. You don't autonomic loops in this SNS and PNS, right? Okay, so notice. Okay, know the structure. Know that autonomic and somatic are different. Autonomic have a two-part party going on. So they have the pre-ganglionic and the post-ganglionic neurons. Okay, neurotransmitters as a pre-ganglionic to post-ganglionic synapse is always acetylcholine, especially for like between parasympathetic and sympathetic. It's always acetylcholine. Whereas at the end here in PNS, it's always acetylcholine, but in SNS, it's mostly norepinephrine. Okay, sometimes there's acetylcholine there too, but it's usually norepinephrine. That's important. Okay. So know the neurons, pre-ganglionic, post-ganglionic. Know where they reside for the most part as well. Okay. More pictures of that. So the sympathetic nervous system. It's usually it's called the thoracolumbar division. Okay. So the pre-ganglionic neuron cell bodies are in the core, okay, in the thoracic or lumbar region, whereas all the sympathetics are in the thoracic and lumbar region. 12 T's and the way 2 to 3 L. Okay. So axons enter and exit the anterior root, right? Remember we talked about the anterior root and the posterior root, right, for sensory and motor functions, and how they form one, and that's going to be a mixed nerve. Okay, know that. This picture here, so it's all thoracic region and some lumbar, right, for the most part here. What's important here is all the things that intervenes with, right, innervates, basically, all these different guys. And we talked about the flight or fight, rest and digest response. And so as long as you're able to kind of think of the bear scenario, you'll be able to understand what the sympathetic nervous system and the parasympathetic nervous system can do to all these different organs. Something in the lungs, what's going to happen to your airways in a sympathetic system? Well, it's simple. Open them up, right? You need more oxygen. Okay, and then parasympathetics can do what to your bronchioles? Good. Okay. Something like your GI. What's going to happen with parasympathetic? Open it all up. It's going to what? As far as motility, what's going to happen with parasympathetic nervous system? Speed up. Yeah. More motility. What about the bladder? What is sympathetic going to do to the bladder? You're gonna have basically. Well, bladder's kind of a mixed thing. Basically, you would need to know you'll pee your pants if you're writing from there. Sympathetic's gonna relax the sphincters at your bladder. Okay. You're not gonna have the tension that you normally would. That one's not that important. More so, lungs, heart, eyes are huge. Yeah.
Do we need to know the pre-vertebral ganglia?
You don't need to know their locations, but you should know that that they exist and what component they are a part of as well.
Also what?
The prevertebral ganglia, and then there's also ghosts. Okay, so with this, heart, lungs, eyes are big here too. So what's going to happen to, or what's going to be affected in your eye by sympathetic nervous system? Okay. Your pupil's going to dilate, but what exactly is happening there? Yeah. What's dilating? Like what's actually happening? This goes to the eye section. The iris, right? And there's two different types of muscles in the iris, right? There's the radial, and there's the one that closes. We'll talk about those too. But you can see how those could be mixed into this type of question about sympathetic nervous system. Okay, so the adrenal glands are important too. They're kind of a continuation of the sympathetic nervous system. The medulla, which is the middle part of the adrenal glands, basically is made from the same embryonic tissue as the sympathetic ganglia. So it's not really the extension to another organ. That's kind of like the synaptic end of the sympathetic nervous system. So they themselves, the adrenal glands, release all of your adrenaline or your epinephrine, norepinephrine. Okay? Two of the same things, slightly different chemically. Do mostly the same stuff. You see here, this is a cool picture because it shows exactly the same thing basically. So instead of going to another organ, it basically just goes, you have the connection to the adrenal glands, which are on top of the kidney, and then the epinephrine and norepinephrine are produced in the middle in the medulla, and then those are released into the blood. So they have a wide effect. A little bit of everything is affected. Okay, not just directly by nerves like you find in your iris or you find in your hearts that are direct sympathetic nerves on those actual organs. Here you have it just affecting the adrenal glands, and it's going to put epi and norepi into your capillaries and into the bloodstream for a grander effect, more widespread. Yeah. Parasympathetics is everything's the opposite. Vagus nerve, cranial nerve 10, right? That carries 80% of the outflow. Big old vagus nerve on either side carrying all your parasympathetics. Same kind of thing here. Note the location. Most of it's all up here. There's no thoracic and lumbar regions, are there? There's some sacral parasympathetics, but nothing here where all the sympathetics are. It's all up here at cranial nerve 10, which is going to be higher or lower on the brainstem?
Lower.
Lower on the brainstem. Yeah. Okay, so the neurotransmitters we touched on a little bit before the break, and here too. So you have acetylcholines at all the preganglionic and both, and all PNS postganglionic. Okay, and then norepinephrine in the SNS at the postganglionic region. And here's more pictures of those. Know those pictures. And notice that's what we just talked about: the bear and the couch, sitting on the couch relaxing. Cool.
Would you say there's somewhere for us to be sitting, right?
Yeah.
Just sit there. Someone put a bear, and then put the food there.
Today?
Yeah, like this morning, I guess, if you didn't see it, maybe it's better yet.
No, I didn't see a bear.
Someone did a bear, like, right before you get the first light into the valley. There was a bear.
I didn't see a bear. How's happening over there?
It's just before light.
Yeah, that's pretty much what that all. That's so let me go back over there. So the fight or flight is basically all the stuff on the sympathetic side here, right? So what fight or flight sympathetic nervous system really prepares you for is you're either gonna, fight and you're gonna be all pumped for fighting, or you're gonna fly away, basically run away. So that's all the stuff on the sympathetic side. Versus the other side is basically the opposite. It's all rest and digest. So you're gonna relax. What's that? Oh yeah, what does that stand for? Is that validation? Yeah, that it's basically digestion and defecation. Yeah. It's everything you do on the couch, kind of. You don't really do that stuff on the couch per se, but like when you're hanging out relaxing, that's all the stuff on the left side, parasympathetic. So those balance each other out throughout the day, throughout all the functions of whatever you're doing in life. So PNS is way more active, while you're sleeping. Sympathetic is probably more alert, things in class, hopefully. All right, sensations. So this was all about eyes, ears, nose, throat, all that stuff, right? So there's special senses and general senses, and we broke down the special senses quite a bit. General we didn't spend too much time on. You don't need to know the different mechanisms, rapid adapting, slow adapting, and stuff like that. But I think you asked that earlier today. Yeah, so don't worry about that. Okay. Know there's cold and warm receptors. I want you guys to just know that for life. But I mean, for this class, you don't really need to spend too much time.
So just learn thermal sensations?
Sorry?
So you just want us to know the thermal sensations?
I mean, there's not much to really know, just they're cold and warm receptors. And then, dermis and epidermis, that's fine. Yeah. Okay. Pain sensations, just fast and slow, all that. Then special senses, these are more important. olfaction, you need to break them all down. You need to know them all pretty well. There'll be questions from all of them. Okay. So olfactory epithelium, up in the upper part of the nasal cavity, there's three different cell types. There's olfactory receptors, there's basal stem cells, there's also supporting cells as well. Okay, know the function of each of them. Then you also have glands there for moisture, and they serve as a solvent for different compounds entering your nasal cavity and help your nose smell better. Okay, so how does it work? There's the cribriform plate here. All these nerve endings up in the very top of your cavity. Look how big it is, way up there. You can only probably get your finger to like right here, and then it hooks back. So it's hard to get back. Like you can't really go back there. You know the structure here. So you have all that epithelium, and then you have the plate, which is the bone there that separates basically your brain from your face. Lightly put. So on this guy. Okay, so you see how this guy here, see how like this is pretty much what the brain is, right? And then you see here, you see all those nerve endings, and they come to the olfactory bulb here, right? That big old blob. Remember on here, you see that big old blob there? The olfactory bulbs on either side, which you should be able to recognize. That's cranial nerve 1. So that has these hairy little extensions through this bone, which is right under here. Right, this bone here, and those are what smell, and it just climbs right up through the bone there to the other side where you have that big bulb, and then that goes back into your smelling, parts of your brain. But those bulbs are where all those endings are pretty much on the other side of your nose. So if you stick your finger high enough up there, you can't touch your brain, but you can be really intimate with your brain, really close by, right on the other side of the bone there. Okay. So know that structure, and know that's where the olfactory bulb is, and then that nerve carries on, and you can see it tracking on that brain model right down to wherever it spreads off into, the unknown inside of your brain. Okay, taste. Know about taste, how it works. So you have two different types of papillae. The fungiform papillae, which are all over your tongue. You have the dilated papillae, and this kind of upside down V area up here, way at the back of your tongue. If you look at your tongue in the mirror, you can see really far back. Sometimes you see the little bumps in the back. A lot of times people think they got like tongue herpes or something. It's not. Everyone has them. Some people you can see them, some people can't. Those are mostly for taste. Because on top of those are where the taste buds are. All right. And the taste buds aren't just on your tongue. You also have them on the roof of your mouth, pharynx, epiglottis as well. Okay, back of your throat. And these papillae are just elevations. Okay, and they have the taste buds. And then here you've got the structure and how it really works. Okay, you have hair cells here. You also have hair cells here for sensation. These are hair cells too. Cilia. Okay. So projections of the cell. Those are what pick up all of the different olfactions. Okay. These pick up taste things, right? So there's three types of cells here as well: supporting, gustation, and basal cells, kind of like in the nose. So how does taste go? You have stimulation of the receptors. Okay, just know the order of this. Okay, so three different cranial nerves partake in taste. The anterior 2/3 of your tongue and the posterior 1/3 of your tongue are different cranial nerves. You don't need to know which ones, but I believe the anterior 2/3 is by the chordae tympani, which is a division of your facial nerve. But you don't need to know that. Just know there's a couple different cranial nerves that carry first order gustatory neurons that innervate the taste buds. The vagus in the back of the throat is where it really works with taste. But there's not much of taste back there. You can't taste heavily in the back of your throat. But a little bit. And it carries up to where? All these different areas. That's why you can have different, you taste something and sometimes you remember something from your past. Or same with smell too. There's all these cross-linkings in your brain. So, I remember one day I was I don't know what I was in, sitting somewhere and I smelled something and it reminded me of my kindergarten classroom. It was like the weirdest thing ever. You start to remember all these weird things. It's because you have all these neurons are crossing this way, that way. An excitatory neuron, might trigger 3 different neurons, and one of those might be connected in 2 different places to other memories and things that are stored in your brain somewhere. So you can have, something triggered just by a smell or a taste. And it's because of all these interlinks in the limbic system, the hypothalamus, and thalamus, and hypothalamus. Okay, eye stuff. So know what all these guys do. These are pretty straightforward. There's 6 muscles. We talked about those in the lab, so that's more of a lab question. I'll expect you to know where they are when we get in the lab section. You should be able to tell me which way they move. More so the easier ones, like lateral rectus. You know it's going to move your eye laterally. We didn't talk about innervation on those really, but just know the way that can make an eye move, okay? When you have the obliques, you can have them move in an oblique fashion. There's also medial rectus and the inferior as well. Okay, then there's layers of the eyeball. You should know these, okay? There's the outer crow. You should know what pointy it does, where it's at. You should be able to point it out. Same with the sclera, okay, and the conjunctiva, pink diapers, okay? Vascular tunic, know all these different things too. You should know that ciliary process secretes fluid called aqueous humor. You should be able to tell me where the aqueous humor is. This is a fancy picture of those. Somewhere here. So there's one here broke at least. And here, all those things are labeled. You ever tell me where all these things are? That's the anatomy part of this. And the eye does have a lot of them, but that's okay. So we have the radial muscles here, okay, and the circular muscles. Know how they work. So what's gonna, it already says on there, but parasympathetic is gonna make the circular muscles work, right? And close the aperture a little bit, okay? Where the radial is just gonna blow everything apart. That's gonna be sympathetic, taking it more light. Retina. So you have 2 layers, neural layer and pigmented layer, okay? Know the structures of these. So light hits which of these first? Without looking at like a diagram. Can you picture the diagram in your head? So which one is it hitting first of these? Which one? So bipolar is in the middle. Remember, it's the ganglion layer. So look at the picture. Ganglion layer here, right? The bipolar and the photoreceptor. So which one's lighting first? No, the light actually hits, remember we talked about this last time, hits the ganglion cells first. It travels past all of those to the back of the retina. Remember that's what we're saying, light's kind of funky. The eyes are kind of weird. So it goes through the ganglion cells, through the bipolar cells, and hits the back of the retina where it activates the photoreceptors, which then send the signals by way of action potentials back to the bipolar cells, not back to, but to the bipolar cells, and to the ganglion cells, which convene at the optic nerve area where they shoot out at the optic disc as the optic nerve. Does that make sense?
It's just going all the way through the back and then all the way through the top.
Yeah, because this here is just a blown up portion of, so this is cut right here, right? Makes it bigger, right? You see this section here?
Yeah.
This is all this. So this here, this imaginary kind of thing, is a retina on the back, the purple thing. So light comes into your eye this way, meaning it's going to come in this way. So it hits here first, but it passes through. So these are exposed to light first, but it passes through and comes back. And the message comes back just a tad bit, and then it all flows on the edge here of the retina, the anterior portion of the retina, back to the optic nerve, which all those ganglion form the optic nerve. It's kind of a tricky question.
It's kind of like a mirror, like the light goes in and bounces back. Goes in and tells her I would see and then bounces back.
No, but that's, you can think of it that way if it helps you, but the mirror is just a reflection of light. So the things you're seeing in a mirror are doing the same thing as this light. This light isn't necessarily like a light bulb. It's any light waves. Like everything we see is because it's bouncing back light waves, right? That's how you can see stuff, right? Like the reason I can see it's red is because it's emitting a certain, the sun's light is hitting and it's bouncing off or reflecting a certain light at my eyes, and that's going to go in through my eye, to the back, past all these guys, and then these guys are going to start lighting up and passing the signal back here and then out. Rods and cones, know the differences, okay? Know where they're situated mostly, know how they work, okay? Know how they how they go. You could have a question. I could have, which order are these going in? And there could be a bunch of different ones with different orders. You should be able to pick out which orders, right? More of the eye stuff. So here's the funny picture, the good picture. So visual consumers in the back, visual consumers in the front. Be able to point out all these things again, eye anatomy, stuff in the first picture as well. Image formation, don't worry about it too much. You should know how it works, but yeah, it's upside down here. But more so than that, and we talked about accommodation on these two. Don't be too concerned with that. More so, know the visual pathway. I really want you guys to know that. And if an image is, if I show you something and tell you an image is here, and then and there's a lesion here, which part are you going to be able to see? Like what portion of your eye are you able to see that? Or if the lesion's back here, like what is that? And this picture is a really good one because you can track it all back. Okay, be able to draw this out. This is going to come up in more in depth in your next set of physiology classes because this is mostly physiology. But if you can do it now, it's better. So be able to draw this out, do some practice ones, like practice where these lesions are. Okay. What if you have a lesion on your left optic nerve? Well, what does that mean? The left optic nerve. That means you can't see these two strands, right? So this purple one is that thing, okay, and this green one is that thing. So you can't see basically the left half of the visual field in each eye. So if you're looking straight, you're not going to be able to see stuff over here. You're not going to be able to see stuff kind of just to the right of your nose. Those are the left visual fields of both eyes, right? Because both of those come in at opposite side, and then it tracks here. Okay. This one here too comes in over here. That's how your eyes see. So be able to tell me that. Now, if there's a lesion here somewhere, what's going on then? That's going to be, well, if the lesion's over here, because remember there's some crossing too. The outer stuff stays, but the middle one's crossed. So essentially over here, if you get a cut here somewhere, then that means you're not going to see anything from your right eye. The turquoise colored stuff that all comes over here is all. So be able to draw those out, practice what lesions are, be able to tell yourself where and what lesion you might see. And then ear stuff, internal, middle, external. Know them. Know the three bones. We're not doing balance stuff. We never kind of really end up touching up on it. That's okay. It's like 1 or 2 sections. You can read on your own. I'm not going to push you on it or ask you anything about it. Know the physio hearing stuff. Okay, how it kind of flows, how you hear stuff. Yeah, equilibrium. Don't worry about this. Thanks. Okay, hormone stuff. This is a little more conceptual. So know how these hormones work. They go into the bloodstream or wherever. So how they can potentially be altered, what their functions are. If there's extra of the hormones, what is that going to do? If there's less of that hormone, what is that going to do? Okay. Here's where most of the hormones I'm talking about are. Hypothalamus, pituitary gland, pretty important. Most of the hormones, the signals are sent from hypothalamus, pituitary, and the anterior, where a bunch of them are made. That's where they get made and they get sent out. Posterior, they're actually made up in the hypothalamus, but they come down to the nerves and they're released by way of posterior pituitary. Okay, know the anatomy of it. Then you can do their stalk connecting the two sections here. See it? And over here, there's the hypothalamus, stalk, and then pituitary gland, about the size of a pea or so. So something so small has so much effect on your body, it's kind of weird. Okay, so then we talked about a few of the different hormones, know what they do. know like something like TSH here. It's stimulated. It's made in the anterior, pituitary, but it's stimulated by thyrotropin releasing hormone from the hypothalamus. And how does that work? Well, the hypothalamus is sensing how much thyroid, T4 you have in your body. So, if your T4 levels are low, then the TRH is going to be released from the hypothalamus into the anterior pituitary. Anterior pituitary will then make TSH, which will go to your thyroids and tell your thyroid to make more of the thyroid hormone. So you know those kind of loops, okay? Involves a little bit of thinking. FSH, LH, we didn't talk about them too much. It's kind of where they went, what they affect, okay? So those are going to affect the testes and the ovaries. And we'll talk about those in the pregnancy. Prolactin, milk production. The rest of them too. Don't worry about them. Melanocyte stimulating hormone. No one knows what it is. Here's a good chart. Kind of shows you what everything does. Okay. Nice and easy flow chart. Know it. Okay, in the posterior pituitary, you have two. You have ADH and oxytocin. Oxytocin is not that exciting. It's just mainly for lactation and smooth muscle contraction in the uterus right after pregnancy, pretty much. Causes it kind of tense up the uterus so it doesn't bleed out. ADH, decrease urine production of kidneys. Okay, if you're not peeing as much, anti-diuretic. All right. And it varies based on osmotic pressure, okay, and blood volume as well. And remember, where are these made? Synthesized in the hypothalamus, carried down to the pituitary gland. That's the difference between the posterior pituitary and anterior. Anterior has kind of an effect by way of messages from the hypothalamus. Posterior is kind of more, reliant on the hypothalamus to actually make those hormones. And here's the feedback system for anti-diuretic hormone. You should know that. Thyroid glands, know the anatomy of it. Pointed out, the two hormones made, T4, T3, and it also makes calcitonin. That's important by way of parafollicular cells. So calcitonin does what? Remember the little thing I told you guys? I remember that. Calcitone down the calcium, right? Calcitonin, tone down the calcium. So the other hormone, which we'll come to in a minute, is from the parathyroid, which is going to be parathyroid hormone to boost the calcium. So this is the guy that kind of counteracts that because you have to have balance. Okay, so thyroid hormones do a lot of stuff. We discussed that in class, right? Everything. I mean, just if you have a lot of thyroid hormones, you can do more. Just think of a person who's like panicking all over the place, sweating, running, jumping, lots of O2, lots of protein being made, that kind of stuff. And then you should know that, like if there's a bunch of T4 hanging out, it's going to go and negatively affect the hypothalamus and pituitary. But if there's low amounts of this, that means TRH is going to be released, and then that's going to go to the pituitary, which is going to release TSH. TSH will come to the thyroid and try to produce more of that. Okay? And calcitonin, again, tones down calcium, inhibits osteoclasts, which then makes sense, right? Because osteoclasts are doing what? They're breaking apart the bone. If they're breaking out bone, there's more calcium going into the blood. So if they're being stopped from doing that, then there's less calcium going into the blood. Parathyroid glands, again, those are the guys that are going to counteract that, okay? There's the chief cells that release parathyroid hormone, okay? And basically regulation of calcium, for the most part, okay? And the parathyroid hormone is going to do what? It's going to increase the number and activity of osteoclasts. So it's going to do what opposite of what calcitonin does, okay? And at the same time, it's also going to slow the rate of excretion from the kidneys. Don't worry about the mechanism for that, just know that it does that, okay? And it forms calcitrol in the kidneys. Calcitrol goes over to the GI tract and tells the GI tract, hey, take in more calcium, magnesium, phosphate as well. Overall, it's going to boost the amount of calcium. Opposite of calcitonin, which decreases. Say like 5 times, just so you can make sure you know it. And here's the cool picture for that. Know it. Pancreas, alpha cells, beta cells secrete insulin. Alpha cells secrete glucagon, know how they work. Good picture for that, we'll tell you everything about that. You can be given a scenario where someone might have really, really low blood sugar. Well, what do you expect? There's going to be more glucagon, more insulin. Or say you have a diabetic patient who didn't eat a meal. Which of those hormones get more prominent in their body? What's going to be kind of their status there? What if someone was like, oh, then you should take some insulin? What is that going to do then? You should do it tell them. And you can kind of figure that all out just from this when we talk about this one. So know this. Adrenal glands, the cortex. Remember, what's in the medulla? Now we're talking about the outside. We talked about the inside already. The adrenal medulla produces, but it's the extension of the sympathetic nervous system, right? That's an example before this. So then that's the inside of the medulla, or the inside of the adrenal gland. The outside is the cortex. It has 3 layers, okay? Mineralocorticoids, corticoids, androgens. Remember, the deeper you get, the sweeter it gets, right? Okay, so mineralocorticoids, aldosterone. Okay, that's sodium, potassium regulation, salt stuff. Salty, then it gets to sugars, then it gets to the sex hormones, right? Okay, so blood pressure and volume. So know this pathway, or kind of the chain of what happens. We talked about this before too, but know about angiotensinogen in the liver, okay, and how it makes Ang1, and it goes to lungs where ACE turns into Ang2, okay, and then that affects aldosterone secretion from the cortex of the adrenal gland. And what is the aldosterone going to do? It's going to come over here and do all this fancy stuff, and ideally pretty much raise the blood pressure, raise the blood volume. What else does Ang2 do? Next lecture, vessels. Remember, we talked about how in low blood volumes, that all your vessels are going to start to constrict to bring that pressure up. But it also affects those arteries and arterioles, and it causes vasoconstriction. That way, you have an increase in blood pressure. And it also goes and affects the pituitary posterior, okay, by way of hypothalamus, and causes more ADH to be secreted. And what does ADH do? Antidiuretic. You're not peeing as much. You're retaining more fluid. Retaining more fluid from the blood section means you're going to boost the blood volume, and then you're going to have higher pressures. Okay. So again, all this blood pressure, volume stuff, it all comes back, and it's all related. And it all starts with renin from the kidneys. Because the kidneys are constantly monitoring how much blood they're getting. If they're not getting that much blood, they're going to start, producing a lot more renin, which is going to go and turn that angiotensinogen into Ang1. Because hey, where's all the blood at? Since we're not getting that much, that must mean we're low on blood. And then all these things are going to happen to bring up the pressure and bring up the amount of blood, or at least the amount of liquid volume in your body. Cortisol does a lot of stuff. Okay, that's a little farther down into the cortex of the adrenal glands. And then the androgens as well. Okay, you have libido and converted estrogens by other tissues, females, and then males is axillary and glucocorticoid. It's significant later. Okay, ovaries, testes, we briefly touched on these. Don't really worry about them. Adrenal glands, melatonin, regulation of sleep stuff. And other hormones, don't worry about those. Either spare the book if you want to know about them, but happy to test them. Okay, so know what blood does. Be able to tell me what all the different layers are? The components of each tube. Alright, so tell me a little bit about what, each of these types as well, all the formed elements, which we talked about and detailed here, right? Again, here's a better breakdown, it's a bit more in depth, don't worry about it too much. But I want you to tell me this guy here, and then you should also be able to tell me, like, if I ask you, what's in the eosinophil, you should be able to tell me that it's a part of what layer it's going to be a part of, right? It's going to be part of your white blood cells, right? Which are going to be up in this little small fluffy coat. These guys don't worry about too much, your proteins. The water's the rest of it, and these solutes are clear. I'm more concerned with this concept. Okay, formation of blood cells, this is just kind of so you guys know where things are coming from. I'm probably not going to ask you a question on any of this, where this is, what does a granulocyte come from? If eosinophil is made by what, a progranulocyte, who cares?
But in lab part, are we gonna have to be able to
Identify the ones I talked about with you guys that day. Be able to identify the structure of the eosinophil, the neutrophil, and those are pretty, they give you a small description in your book on that one page, just pretty much based on nuclei and colors as well, you can tell. Red blood cells, be able to identify them, that kind of stuff. The cycle, tell me about the cycle. There's this kind of little pathway, okay? So, you don't need to know it in great detail, but like if I ask you, what's the red blood cell split into, you should be able to tell me it's hemoglobin, right? You should be able to tell me that, heme has iron as a part of it, okay? And that somewhere along the way, there's excretion of this bilirubin, there's bacteria involved in the production or the excretion process. So I'm not going to ask you like all these big fancy words in intro class. Okay. But like you should know that somewhere along the way in the process of blood being broken down, a lot of it is recycled, but other parts of it are peed out and pooped out. Okay. Okay, know about white blood cells, nothing in too much detail, like just pretty much what's here. Know what the lymphocytes are, B cells, T cells, natural killer cells as well. They're not that cool. They just attack everything. But B cells and T cells are pretty cool and important. Platelets are really important for clotting. Okay. Know about them. Know a little bit about hemostasis. How you have the spasm, you have the platelet plug formation, and you also have this whole clotting cascade. But we're not going to, you don't need to know the entire clotting cascade. But know that you have fibrin, and fibrin's kind of like a big guy that plays a role there, okay? And that's converted from fibrinogen by thrombin, okay? And this here. So you don't need to know all these different factors and stuff, but know that you have this prothrombin turning into thrombin, and then eventually you're going to have the fibrin. Fibrin's what's going to cause your clot to be formed, okay? That's the important part, okay? Okay, know about retraction and repair, general ideas, okay? Know that. I'm not going to ask you to tell me what, much about a pulmonary embolism. But, blood type stuff, we went over this pretty good detail last time. You should know it. There will be questions on it. Like this stuff. Well, a patient comes in type B blood. What kind of blood can they get? What kind of blood can they not get? Okay. The racist stuff, we didn't really go over it in too much detail. We talk about it more in pregnancy, but no, it's also that factor there. Okay, another antigen on the blood cell. You don't need to know this. That's it. All right. Any questions? Comments? Etc.
What are some of the things you can expect during
Oh yeah, I knew I was getting something. That's blood part 2, right? Yeah. Oh man, okay. Blood and circulation stuff. Oh yeah, know the vessels. I mean, that's pretty straightforward. You should know a little bit about each one. If I ask you the differences between arteries and veins, you should tell me some differences. The anatomy of a vessel, okay? How there's 3 layers. Where are you gonna find simple squamous inner endothelium? You should be able to tell me simple squamous. What if I just give you a histological picture of an artery and ask, what is the inner layer? What kind of cells is it? I mean, you already know it's simple squamous. That's, you should know from your notes, but maybe the picture will only help give you a visual clue. Because if you forgot what type it is, you can probably tell just because there's one layer of squamous cells around the vessel. Okay? Know the different artery stuff, sizes, and here's some pictures. Capillaries are the exchange vessels, you should know that stuff, how they work. Veins, venules, same thing, they're low pressure. Okay, and there's 3 different ways you get a return of venous blood, right? Contraction of the heart, skeletal pump, respiratory pump. Which of these is not a way that you get, return of venous blood? You can be a 4th answer there that isn't a part of these ones. Something obscure and weird. High pressure, low elasticity. Okay, let's just pass. So, blood pressure goes high to low. We talked about how that works. That makes sense. In way of physics, right? Contraction, relaxation of the heart is diastole and systole. Or systole is contraction, diastole is relaxation. Okay, so you have your systolic or your diastolic blood pressures, right? And where can we measure blood pressures? Not anatomic, but what type of vessel is best for measuring? Arteries, right? Okay, good. What's an ideal pressure? Yeah. Okay, good. A lot of things dictate, what your pressure is. So here, this is a chart, right? So you're measuring the artery pressure at systole here, where it's pumping, so it's a lot, and when it's relaxing, it drops. Okay, and that's gonna be 120/80, ideally. And then everywhere else it kind of just evens out. That's why you don't get a pulse. You don't hear a change here. So you can put your finger over a vein, like on your hand, you're not gonna get a pulse. Because the pressure in systole and diastole is the same. Okay, so resistance by way of the size of the lumen. Okay, this is kind of river analogy we talked about. Blood viscosity and then the total blood vessel length too. So thicker blood, slower flow, size of lumen. If you narrow it down, you're gonna have more of a resistance there. You're gonna have speedier blood. Your pressure's gonna be higher. As if you open the mouth of the river, slower flow, relaxed, not much pressure there. So these 4 guys are really big in regulation of blood pressure and flow. We talked about heart rate, faster heart rate, more of a pressure coming, because there's more blood being pumped in your vessels, higher pressures. Stroke volume is the amount of blood that your heart pushes. Remember we talked about you can have a slow rate but a huge stroke volume, and you're going to have more coming through. Or you can have a really small stroke volume and a really fast rate, and you're still going to have a low pressure too. That happens in people with some sorts of heart arrhythmias where they're beating really, really fast, but they're not pumping out a lot of blood because they're not opening and closing all the way. And so then the blood's just kind of sitting there, hanging out inside those chambers of the heart. It starts to coagulate, and then you get clots, and they shoot up your brain, you have strokes and stuff like that. Okay, cardiovascular center, we briefly touched on this. There's a couple of different types of receptors. I should be able to tell you kind of what the receptors, how they kind of function, but nothing in too much detail. Hormonal regulation, we've already talked about all these guys in that one picture pretty much, when they're renin, ectin, cytokinin, sinogen, whatever you want to call it. The whole pathway thing, the RAS pathway, know that, okay? And that will cover this whole cycle. Okay, the routes. Systemic, pulmonary, hepatic. We didn't talk about fuel. So these are pretty straightforward. How the pumps and stuff, we do that on the board and talk about it, right? We should be able to kind of trace that out for systemic. We went over all the major arteries. the arms, legs, up to the carotids and things like that. Talk about all the gut ones as well. Those are going to be more so important for your lab exam, but I mean, maybe you'll have one, but it'll be a pretty, you know, say it's something like a carotid, you'll have a big blown up picture of the head, and then you'll have all the vasculature there, or not all the vasculature there, but the main stuff. You know, either I'll ask you before the split or something, it's internal versus external. Be able to identify those things. Net or splash marks. Like something like this. The trunk, we talked about the aorta, how it ascends first, then it descends, comes down, right? All the different branchings of it. And you have the intercostals, then you have the celiac trunk coming off, superior mesenteric artery in the gut, and the inferior mesenteric, right? All these guys. These guys. Veins, we said we're not going to really go crazy over. Pulmonary stuff we talked about, and then the GI kind of detour a little bit with the hepatic portal vein coming from the gut, right? It comes from two different sections. The splenic vein and then the superior mesenteric vein forms the hepatic portal veins. So it brings all the nutrients from the gut into the liver where they're processed, right? So that's like the only vein you kind of need to know. So that's the only vein, if I asked you anything about it, would be this one, the hepatic portal vein. So where does the hepatic portal vein come from? It comes from those two guys, but what is the purpose of it? Would be something I would ask. If anything else, that's what I'm ask you. What's the purpose of it? Nutrients. Bring nutrients from the gut into the liver for processing. So you can do, with people that have liver failure, so the liver's all messed up, nasty, you can shunt away, because the liver's getting blood from the arterial system too, and this system. You don't want it to get overwhelmed. People with failing livers, they can shunt this blood away. Get in there, chop it up, cut it off, put it somewhere else. Less stress on the liver. Okay, checking circulation stuff, we can do that now. And that's